Good morning, everyone, and welcome to the February webinar of the St. John Naturalists Club. I'm Julie Bauer, the program coordinator for the club. So today we are having guest speaker Dr. Alfredo Justo, the curator of botany and mycology at the New Brunswick Museum, and he's going to be speaking on the biodiversity of chanterelles and hedgehog mushrooms in New Brunswick. But before we begin, I just want to speak a little bit about our club. So we are a growing community of naturalists in the province and in St. John area. And we're always looking for new members. We have individual membership and family membership. We're pretty active. We're going to be getting even more active as the spring comes. Coming up on the 26th of February is winter birding for beginners, and that's going to be at Rockwood Park. It was originally going to be tomorrow, but the weather and the trail conditions weren't too great, so we decided that we're going to move it to the 26th. So the registration's required, and we'd love to have you join us. After that, we're also doing another winter birding on March 8th at Dutch Point Park in Hampton. And so we welcome everyone who is a beginner, or you can be already experienced. We're going to provide binoculars if you need them. And then coming up next month is the Point Lepreau Bird Observatory Volunteer Training Workshop. So if you're not familiar with the Point Lepreau Bird Observatory, it's a project where we monitor the migrating seabirds and sea ducks that go past the point in the spring and in the fall. And so if you're interested, you can contact us or come and attend the workshop. So we'll teach you all that you need to know. It's on March 26th at Rockwood Park Interpretation Center. Another announcement is the Shorebird Conservation and Education Project is looking for more committee members. So if you are familiar with the project or not, it's a project that takes place at Irving Nature Park. We're looking to expand to other beaches, but it's basically an outreach program that educates beach walkers on the importance of allowing the shorebirds that are resting on the beach to allow them to rest before their migration, and that happens in late summer. So if you're interested in being on the committee, we only meet twice a year, and it would be for a one-year term. So just get in contact with us. Here is all of our social medias and our website. So check us out and follow us. But I'm going to pass it over to Mary, who's going to introduce Alfredo.
Hey, well, thank you, Julie, and good morning, everyone. And before we get started, I just would like to ask everyone to type your questions that you have for our guest speaker into the chat during the presentation, and then Alfredo will address those questions when he's done his presentation. So today's guest speaker is a familiar face to our naturalist community. He's had many presentations and led many guided mushroom outings. Dr. Alfredo Justo is the curator of botany and mycology at the New Brunswick Museum. He received his PhD in systematic mycology at the University of Vigo, Spain, in 2006, and then worked on mycological conservation and diversity projects in Spain. Alfredo went on to do a postdoctoral research position with Clark University in Massachusetts, where he focused on molecular systematics of mushroom-forming fungi. He then held research and teaching positions in Mexico, Spain, and eventually back to the USA, where he worked as a visiting assistant professor in the biology department at Worcester State University and as a visiting scholar at Clark University. Well, Alfredo then made his way to his current home here in St. John, New Brunswick, where he joined the Natural History Department of the New Brunswick Museum in 2019, just pre-COVID. He oversees the curation and growth of the New Brunswick Museum Herbarium, which comprises over 100,000 specimens of plants, fungi, lichens, bryophytes, and algae from New Brunswick and neighboring regions. Alfredo's current research focuses on the fungal diversity of New Brunswick and Atlantic Canada, and he maintains an ongoing global taxonomic research on selected genera of mushrooms. So with that introduction, it's my pleasure to welcome Dr. Alfredo Justo, and I will pass the camera and the microphone on to you now, Alfredo.
Thank you, Mary, for the introduction, and thank you to the St. John Naturalist Club for inviting me to give this presentation. I'm going to start checking that everything works correctly with the sharing of my screen. Yeah, making sure everything is there. Okay, so I'm going to tell you a little bit today about the biodiversity of two groups of mushrooms that are really popular as edibles all around the world, Chanterelles and Hedgehog mushrooms. And the study that we did back in 2021 that in a way is still ongoing about the diversity of these groups in the province. As Mary mentioned, I'm the curator of botany and mycology at the New Brunswick Museum. And what I'm presenting today is part of our daily research activities at the museum. There is a lot of the museum that is public facing, but there is a lot going under the scenes, and the research that happens every day is one of those things. And also I wanted to mention that even though I'm going to be the 1% in the results, this type of work cannot be done by one person alone. And the fantastic team that we have in botany and mycology, you see them here in the photo, Amanda Brebner, Kendra Driscoll, and Alex Hood, that were fundamental to carry out the research they're presenting today. Just a couple of slides just to give you a bit of background of what the New Brunswick Museum entails in terms of mycology and botany in general. The NBM herbarium has over 100,000 specimens, and the purpose of the herbarium is to document the species that we have of plants and fungi. My predecessor in the position, Stephen Clayton, was a lichenologist, and thanks to him and many collaborators, we have probably the best lichen collection in Atlantic Canada. The purpose of the museum is also, once we have gathered that knowledge, is to spread it out through activities like this one, where we tell the general public about our finds, about the diversity of plants, fungi, animals, fossils, anything that you can imagine. We have to document it, and then we have to kind of spread out that knowledge through different programs, both inside of the museum, but also external programs. As I mentioned, we have over 100,000 specimens in the New Brunswick Museum herbarium, but those specimens are not evenly distributed according to taxonomic group. And this is a comparison that I always like to make, because the most familiar organisms when you talk about botany and mycology are usually plants. And we have quite a good collection of plants, of New Brunswick plants at the museum. We have over 42,000 collections currently. The collection is still growing, and that documents about 1,600 species of plants that we have in New Brunswick. We still find interesting records and new things every year, but we have a pretty good idea of the plants that occur in the province. When it comes to macrofungi, we actually estimate that the diversity is at least double that of plants. We have probably around 3,000 species of macrofungi, and by this I mean fungi that actually produce things, mushrooms that we can see. We're not even counting the microscopic fungi that never fruit and never produce mushrooms. We have at least 3,000 species, so double the number of plants. But we have less than 10,000 collections currently at the New Brunswick Museum herbarium. So that's the part of the museum collections that I'm trying to grow. That is really the focus of my research program at the museum, is to grow the mycology part of the collection. And when it comes to mushrooms, when it comes to macrofungi, it's sometimes very hard to know where to start. They're so diverse, there are so many species, so many different groups. It's hard sometimes to decide how to approach the studies of macrofungal diversity. And you have examples here. Most of the photos, or all of the photos that you're going to see today, unless stated otherwise, are of New Brunswick mushrooms, are of New Brunswick collections that are currently part of the New Brunswick Museum herbarium, but the diversity is so high. There are so many different groups, and you need so many different taxonomic expertises to kind of try to tackle the different groups. That is kind of a daunting task when you think about the road that you have ahead of you to document all that diversity. So one of the projects that we selected to be one of our first projects to document fungal diversity focuses on two very charismatic groups of mushrooms, chanterelles and hedgehog mushrooms. You see chanterelles here, the golden chanterelles that most people know and collect. We also have trumpets like the genus Craterellus, which is related to chanterelles, and we have hedgehog mushrooms, the genus Hydnum, which are also very popular, and actually, even though they look quite different, they're very closely related to chanterelles. So why did we select this group? Well, to begin with, there were very few collections of chanterelles and hedgehog mushrooms at the New Brunswick Museum herbarium when we started this project. There were less than 50 collections. And none of those collections had molecular data, DNA data. In this day and age, any fungal diversity project needs a DNA component to start figuring out the diversity that you have. Mushrooms are very, in many cases, in many groups, they're very cryptic. They don't have a lot of morphological characters that are going to tell you right away who they are. So you need the help of DNA to try to figure that out. Any fungal diversity project these days has a DNA component. So in 2021, we applied for a research grant for the New Brunswick Wildlife Trust Fund that funds biodiversity-related projects in the province. If you want to contribute to the Wildlife Trust Fund, you just have to get in your car this license plate with the conservation sign, and the small fee, extra fee that you pay goes directly into the New Brunswick Wildlife Trust Fund. So the Trust Fund gave us a one-year project that basically covered the 2021 field season to collect new collections of chanterelles and hedgehogs, and then a few months after that to kind of process all the data. So the goals for this project was to go out weekly. We focus mostly in the southern part of the province because we wanted to be able to go out for a full day of collecting and be able to come back to the museum to process the collections the next day. One thing when you're working with macrofungi, you don't have a lot of time after collecting to get the collections through the dryer, to process them for long-term preservation of the collection. So we focus mostly on the southern part of the province. And we also put out a call through social media for anybody, any of the non-professional mycologists that are in the province. Just if they find anything interesting, please send it to us and we will include it in the project. And as you will see, some of the collections, some of the interesting records came through that general call. And we set a target of at least 200 new collections of chanterelles and hedgehogs. After the collecting was done, what we need to do is put all the pieces of the puzzle together. We look at the mushrooms in the field, we record the habitat where they're growing, we look at the external morphological characters. In the lab, we're going to put them under the microscope and look at characters that are important. For this particular group, mostly spores, size and shape and other things. These fantastic photos of microscopy are not mine. They're from Dave Mallock, one of our research associates at the museum, who does fantastic microscopy of mushrooms. We also, again, look at the ecology. Was this mushroom growing under conifers? Was it growing under hardwoods? And then after the collecting is done, we generate DNA data. And at the end of the day, mushroom taxonomy is putting all the pieces together. And what does it mean? What does the morphological variation mean, the ecological variation, and the DNA variation? How do you fit all together into a story that makes sense? We set up an iNaturalist project page that automatically captures any observation of Hydnum, Cantharellus, or Craterellus in the province, whether it's part directly of the project or not, is going to get captured in the project, and in the future is a good way to follow how many new records we add to the knowledge of these groups in the province. So what were the results? So 2021 was an excellent mushroom year for everything, but certainly for chanterelles and hedgehogs. So we were able to collect more collections that we have initially planned for. We collected 277 collections, about 160 something of Hydnum, and then the rest was Cantharellus and Craterellus. And again, all the photos that you're going to be seeing throughout the presentation are collections that were made during the project and are currently part of the New Brunswick Museum herbarium. Let's start with hedgehogs, because as you're going to see, are the more diverse and probably the more interesting group. There are 5 species of Hydnum that are commonly or were commonly reported in North America: Hydnum albidum, albomagnum, repandum, rufescens, and umbilicatum. The species that you see here highlighted in a different color, we know they are in North America because they were originally described from North America, so they are North American species. These other 2 are actually really commonly used names for species of Hydnum, that these were originally described from Europe. And you're going to see in a minute, in many cases, those species are not present in North America. We have similarly looking species, but they are not the same ones. One of the reasons we wanted to do Hydnum is because we had reference data to look at. So when we generated our own DNA data and our own collection data, we could look at reference works that have looked at Hydnum in North America. There is the work by Niskanen and collaborators that was out 2018 that was looking diversity in North America and Europe, and especially the monograph by Rachel Sweeney and collaborators that was also out in 2018 that look specifically at hedgehog in Eastern North America. What species do we have? What are the correct names for the species that we have here? So we could compare our data to the data that was already out there. This is I'm gonna go kind of point by point here, but just a general overview of the results of the DNA study. We were able to sequence about 134 collections of Hydnum from New Brunswick. And the overall result is that we have 13 species of Hydnum in New Brunswick. 2 of them, we already knew they were here. There were previous confirmed records of the species here. 7 of them were new records for the province. We had the first full confirmation that this species was here. 3 of them were new records for Canada. At the moment we collected them, they had not been recorded anywhere else in Canada. And 1 was actually an undescribed species that very soon will be formally described. And the colors that you see in the DNA study signify the 4 different groups within Hydnum, within hedgehogs. There are subgenera within the genus Hydnum, and they're usually well-defined both by morphology and by DNA data. So when I'm presenting the different species, I'm going to follow kind of the order of the subgenera, because the similar species, similarly looking species, are going to be grouped together there. Let's start with subgenus alba. As you can imagine from that name, that means white. It's usually the species that have white colors or very, very pale colors. This is one of the species that was described originally from North America, Hydnum albidum. Hydnum albidum has completely white or extremely pale forms, extremely pale mushrooms. It only stains a little bit orange, but it takes a while to stain. So for the most part, in the field, when you find it, it's going to be mostly white. And we had a few records of Hydnum albidum in the southern part of the province. We also found what was a first record for Canada at the time, which is Hydnum albuarantiacum. It's not surprising that it was a first record for Canada. This was only described in 2018 in the monograph that I mentioned before for Eastern North America. So Hydnum albuarantiacum, the name makes reference to the fact that the mushrooms are mostly white, but they stain orange very quickly, and as soon as you touch them, they're going to stain quite bright orange. And also, if it rains a lot, if something falls into the mushroom, you're gonna see the mushroom staining pretty fast. Another subgenus related to subgenus alba, subgenus pallida, and here we have one species, Hydnum subtilior, was a new record for New Brunswick. Subtilior usually has pale colors, but it's more intensely colored than the species that we just saw. It stains orange pretty brightly, even in the stipe, which is not common for many species of Hydnum. And the length of the stipe is usually much higher, has a very long stem compared to the diameter of the cap. The other group of hedgehogs is the species that mostly we were going under the name Hydnum repandum, which is still a name that you're going to see around in a lot of field guides, in a lot of web pages, but Hydnum repandum is a European species. What we have in North America and what we have in New Brunswick are similarly looking species, but they are particular to this continent. And you can see by far the most common one is going to be Hydnum washingtonianum. At the moment of the description was a new record for New Brunswick. Hydnum subolympicum, which looks similar, but is slightly different. And I want to mention Hydnum vagabundum, that it was a new record for Canada. Unfortunately, we don't have a photo for this, but this was one of the collections that came from people outside of the museum. This is a collection from Anthony Brooks, from Michael MB, that was made in Odell Park. We don't have a photo for it, but we have the collection and we know the species is in the province. This is the most common Hydnum repandum-like thing that we have in the province, Hydnum washingtonianum. The name Washingtonianum refers to Washington State on the West Coast. And this is a species that mostly occurs on the West Coast for North America and the East Coast of North America. And this is a very big, bulky species. The cap can be well over 10, 15 centimeters in diameter. And it changes color quite a bit as it grows. These are young specimens of Hydnum washingtonianum. And you can see this pale peach orange color. They get much paler with age. The thing about Hydnum is that they remained in the field for a really, really long time. They don't decay pretty fast. So with rain and with staying in the field for that long, they usually get much paler in color if you find them quite later after they fruit. And this is another species of the same group, but it's much rarer, at least based on what we know so far in the province. By far, Washingtonianum seems to be the most common of the big Hydnums that we have. This is Hydnum subolympicum that was described originally from Newfoundland. And it's similar in color to Hydnum washingtonianum. It has kind of a slightly different appearance. It's more, the stipe tends to be more elongated. And one thing that Hydnum subolympicum does that you can see here in the photos is the surface of the cap tends to crack. Those cracks seem to be very typical of Hydnum subolympicum. The spores, if you put them under the microscope, also will help you separate subolympicum from washingtonianum. But so far, we only have two collections of subolympicum in the province. One is from the Fundy Trail Parkway, and the other one is from Spetnik Lake. And then we get to the most complicated part of Hydnum, the hedgehogs, the small, bright orange, kind of brownish color hedgehogs. This is the subgenus Rufescentia. These usually also stain orange as you touch them. And here we have 7 species. This is where most of the species diversity is going to be present. And I group them kind of arbitrarily and subjectively in three species that are kind of easy to recognize, that I think after looking at these things more in detail, I have a good feel for how they look out there in the field. And four that are going to be extremely difficult to separate unless you have sequence data, unless you have DNA data. So let's start with the relatively easy ones. This is Hydnum aerostatisporum. It was a species that was described from the southern United States, but we have it also up here in Canada. These photos that you see here, they're not actually from 2021. These are from 2022 from the Biota project up in Kennedy Lakes. As you know, the New Brunswick Museum has the Biota program where we go out for 2 weeks somewhere in the province and document the diversity of everything there, mushrooms, animals, everything. And these species, I identified them in the field as Hydnum aerostatisporum, and we just got the DNA data back 2 weeks ago, and they're actually Hydnum aerostatisporum. So yes, it's one of the species that if you start paying attention to the morphological characters, you can probably recognize in the field, and you can see this species to me in the field, it really looks much bulkier than any of the species in subgenus Rufescentia. It tends to have a long stem, and the cap in age tends to develop these small squamules. The surface is not completely smooth. So I think once you get familiar, if you have the species in your area, once you get familiar with it, it's going to be one of the ones that you can be relatively confident to identify based on morphology. This is Hydnum quebecense. Quebecense was one of the species that was also described in the studies that I mentioned before. Quebecense was given this name because the original material comes from Quebec. So we kind of suspected that this was in the province. And if you look at where it's growing, it's going to give you one key character that is going to be important for identification. This grows mostly in bryophyte cover soil. In the original description, they say sphagnum, but it doesn't have to be sphagnum. It can be basania, it can be other bryophytes that cover the soil in very humid areas. And it has these very bright kind of orange-red colors in the cap. So with the habitat and the colors of the cap, you can already tell, you kind of get a sense of that you're looking at Hydnum quebecense. If you use the identification key that that is present in the work by Rachel Sweeney and collaborators, you reach a point in the dichotomous key where it's going to ask you where the Hydnum is growing. Is it growing in a sphagnum, or is it growing directly on soil? As I mentioned, it doesn't have to be a sphagnum, but this is a good character. Is it growing on moss bryophyte covered soil, or is it growing just on bare soil? And if it's growing with bryophytes, you're going to reach a point in the key that is going to ask you about the spore size to differentiate between Hydnum quebecense and the other species. But this was the only species that was left unnamed, undescribed in the work, in the monograph by Rachel. And they gave it the code SPAS30 because that was the collection number, but they couldn't formally describe it because there was only one collection. There were no photos of how the mushroom looked like, and we knew from the DNA data that was different and from the morphology, but there was no way to describe this. And during our research in a research project in New Brunswick, we found over 30 collections of these species that Rachel had left undescribed. We have this is the collection that was originally included there. It was from New York in the States, and all the rest are New Brunswick collections. And you can see that they were really correct in pointing out this grows in the same habitats as Hydnum quebecense. This grows in moss-covered soil, be it sphagnum, be it some other bryophyte. And you can already tell by looking at the photo that there is one character that is going to be different from Hydnum quebecense, which is the color of the cap. Hydnum, this species, has a much dull brown color than Hydnum quebecense. Hydnum quebecense is very bright in the field. Both species tend to fruit in the same places at the same time. And when we were collecting them in the field, we couldn't put a name on them yet, but we knew we were collecting at least two different species. We were collecting the bright red Hydnums, and we were collecting the brown Hydnums that grew on moss. So we didn't have a name for them yet, but we knew they were different. I'm cheating a little bit here because I'm giving you the most typical perfect photos of these species. There is a little bit of variation in color. It's not always this perfect brown, completely different from quebecense, but for the most part, you're going to be able to identify most of the collections in the field. And this species is, we contacted Rachel Sweeney at the University of Tennessee, and we asked her, well, did you find any more collections of this thing? Because we have it in New Brunswick, and obviously it needs a name. There is actually one more collection from Labrador, from the Newfoundland foray people from Andrews Boyd, that he graciously gave us the collection to include in our study. And currently this species is in the papers, is currently under review, but it's going to be named Hydnum atlanticum for its distribution along the Atlantic provinces and states of the United States. And currently it's only known from New Brunswick, one collection in Labrador, one collection in New York. I don't know how widespread in other parts of Eastern North America it's going to be. Probably it's going to be present in sphagnum-dominated bogs in other areas, but I don't think it's going to really go much further south along the Atlantic coast. So we have a new species of mushroom being described from New Brunswick. And then we get to the last part of Hydnum, which is not a nice part if you like to do taxonomy and if you like to identify all of your collections. Because if you follow the key that Rachel Sweeney prepared, this is the last point of the key. There are 4 species that key out together, and the only information that kind of has to separate them is habitat and distribution, which this information obviously changes over time. If you look at the DNA data, there are clearly different species. There is no doubt that these things are different, but there are very few morphological or ecological characters that are going to allow you to separate them. But we have them all in New Brunswick, so we better get used to kind of using DNA data to try to get a better sense of how they're distributed. So there is Hydnum umbilicatum, and Hydnum umbilicatum is a very common use name for all these small Hydnums, and it's actually quite common. Of these 4 species that I just mentioned, it's actually the most common one. And umbilicatum makes reference to the small depression at the center of the cap, but there are other species that can have that. So that's not always a good character to identify umbilicatum. But again, small brown, orangey brown Hydnums not growing directly on moss, it's probably going to be umbilicatum. But we do have other species like this one. This is Hydnum cuspidatum, which is a new record for Canada. And this was originally described from the southern United States. And we have only one collection of these species. And we sequence most of our small brown Hydnums. And this collection is from the Kingston Peninsula near St. John. And then we have Hydnum submulticolor, these guys here, and Hydnum canadense. Canadense was also given the name because it was originally described from Newfoundland. And again, these 4 species, if you don't have DNA data, it's going to be extremely difficult to tell them apart. But we also have to, again, get used to incorporating that type of data when we do biodiversity studies and biodiversity kind of management of things. So to recap very quickly about hedgehogs in New Brunswick, we have 13 species. 2 of them had been previously recorded, previous to our study of Hydnum. 7 of them were new records for the province. 3 of them were new records for Canada, and there was 1 species that was currently undescribed at the moment of the study, but it is going to be named Hydnum atlanticum very soon. So what about the other guys? What about the chanterelles? Chanterelles is kind of we start with a similar situation like the one that I just described from Hydnum. There is a European name that has been widely used for collections in North America, and that name that you're still going to see around everywhere is Cantharellus cibarius, the golden chanterelle. And there has been a lot of research in the past 10, 15 years about chanterelles, both in Europe and North America, that kind of has clarified a lot of what names can we use for the species that we have here. I'm going to start by saying, before we figure out what we have in North America, most of the names originally come from Europe, so it's good that they figure out the species that are actually present there. There was a big study published in 2016 by Lariaga and collaborators, and they kind of define very clearly what 8 species of Cantharellus are present in Europe. So that's a good reference point for anybody doing work on chanterelles anywhere else in the world, because now we know what the European names mean and how they should be defined. Now, in North America, we have a much higher diversity of chanterelles, not necessarily us, but in general, in North America, there is probably around 30 species of chanterelles, or at least chanterelle names, that have been proposed in recent years. And these are just some examples of recent publications from, and if you see the dates, this is 2016, 2016, this is, I think, 2017, 18. It's very recent how we're starting to tease apart the species that we have. Rachel Sweeney from the University of Tennessee, she was also doing chanterelles for her PhD project, and she just published a fantastic study about chanterelles in the southern US, but it kind of, some of the things that come into view here are of interest for Eastern North America in general. They have much more diversity of chanterelles down south than we do here in New Brunswick. Most of our diversity is going to be here. These 3 groups that you see at the top of the tree, the Cibarius complex, the Tenuithrix complex, and Cantharellus camphoratus. This is where our diversity, at least as we have recorded it so far, is really concentrated. So for chanterelles, for Cantharellus, we sequence about 44 collections. We have one very common species, is the one that you see here in this group of DNA sequences. This is Cantharellus enelensis, and this is Cantharellus cibarius from Europe. So you can see, if you just look at these phylogeny, you see, well, they're different. In the paper that I just mentioned by Rachel Sweeney, it's a little bit of controversy how well you can separate things in the Cibarius complex. But if I were to look only at the collections that we have here, for sure, I would call them Cantharellus enelensis. Then we have Cantharellus deceptivus and Cantharellus camphoratus. So again, this is the more common species that we have. Enelensis, the name was given because it was described from Newfoundland. So NL makes reference to the initials of the province, and is by far the most common chanterelle that we have in the province. It was originally described as only occurring with conifers, but it at least occurs with birch. We have collected these under birch and without any other possible mycorrhizal partners nearby. This is another very interesting record that came from the general public. We didn't collect it, the people at the museum. This comes from Holly Jones, also from Maiko MD. And this is Cantharellus deceptivus. And this is the iNaturalist observation that Holly created and some of the photos that she took. And when we sequenced this, we saw this is part also of another species complex, but for the time being, we're using the name Cantharellus deceptivus. And this is Cantharellus camphoratus, which is a very curious case in fungal diversity. This was originally described from Nova Scotia, but the name was barely used for years and years and years. In the paper that was describing Cantharellus enelensis from Newfoundland, they were able to study the original collection of Cantharellus camphoratus, and they saw that it matches many of the collections they made in Newfoundland. So they kind of redescribe it and bring attention again to the name Cantharellus camphoratus. We also have it in New Brunswick, and it's widespread. It's not particularly common, not as common as Enellensis for sure. And it has a very curious morphology, and it kind of falls into two categories when you find Cantharellus camphoratus. You kind of find these massive fruit bodies, and sometimes it fruits only one, which is rare for chanterelles, they usually fruit in groups. These kind of massive fruit bodies that are probably the consequence of several fruit bodies fusing together, or you can find Cantharellus camphoratus with this morphology, with this very irregular underside of the chanterelle. And again, kind of several fruit bodies kind of fruiting all together without properly separating from each other. And we do have a couple of localities of camphoratus in New Brunswick, and it's a regional species. It makes its way down the East Coast, but it's a species that was originally described from Atlantic Canada. And then the other group that is the group that probably we need to put more effort in future years are the winter chanterelles, or the trumpets. So far of the collections that we did for the project and the collections that we were able to sequence, we have 18 collections that were sequenced. I would say for Cantharellus and Craterellus, molecular work is tricky because you have to find the barcode that we use for most fungi doesn't work well for this group, so you have to look for alternative genes to sequence. And so far we have 2 species based on molecular data, 2 species confirmed, Cantharellus tubiformis, Craterellus tubiformis and Craterellus phallax. But we know we have more species than these 2. It's just a matter of finding collections and studying those collections and confirming that those species are here. So that was the overview for hedgehogs and chanterelles. I hope I got you excited to go out in the field and look for these things this season. And before I finish the talk, I want to give you a preview of all the other stuff. We just sent 1,000 collections from New Brunswick mushrooms from different groups, mostly for our Cortinarius project, but also from different groups to be sequenced. And we start going through the results right now. So it gives you like a snapshot of the initial picture that it starts being defined for many of these groups. This year, the project that we applied for at the New Brunswick Wildlife Trust Fund was to sequence collections of Cortinarius. Cortinarius is the most diverse genus of mushroom-forming fungi. It has been recently separated into several genera and is still the most diverse genus of mushroom-forming fungi. We sent for sequencing about a little over 500 collections. We have data for about 400 now. And we can already confirm we have 150 species, and this is one genus, 150 species of Cortinarius in New Brunswick. 24 of those species don't have a name. They're most likely are undescribed. Many of them are also recorded in the recent catalog in the repertoire des Cortinarius du Québec that it was published in 2021 by our fantastic mycologists that we have next door in Quebec. So we have a lot of reference data from them, and many of our unnamed species are the same ones that they found, and they cannot put a name on them either. But we have a couple of things that they're not even recorded in the study from Quebec, that only known from the collections in New Brunswick so far. So lots of work to be done to clarify things. Waxcaps are another group that we send out for sequencing, and we have data about 50 collections, which is not a lot, but I can tell you already that every single collection that was given a field ID based on morphology using a European name is not the European species. This is a New Brunswick collection of, and these are names that are commonly used everywhere still in Eastern North America, in web pages, field guides, and these are the names that are out there. This is Gliophorus irrigatus, this is not Gliophorus irrigatus in the true sense. It's not a European species. This was named Gliophorus cetacinus, which is again a European species. That's not what we have in New Brunswick. Same for these guys. These two species, Hygrocebi punicea and Hygrocebi splendidissima, they're both European species. They're both species listed as vulnerable in the IUCN Red List for conservation. Morphologically, these look quite similar. Molecular data are telling us these are not the European species. These are something that is related to them, something that falls somewhere near them in the DNA studies, but they're not the same species. So every group of mushrooms that you're going to be looking at, you're going to be finding very similar situations. And the only way to really move forward and proceed with that is just try to generate as many, first of all, collect as much as possible and document collections properly, DNA sequence as much as you can, and then put all the pieces of the puzzle together. There is a homework part of this, then you have to do the taxonomic group. The fact that these things are not the European species doesn't necessarily mean that they're new species. They might be older North American names that are available for these species, but you have to sit down with all the data, go through all the taxonomic information, and try to figure out what species do you have in the province. So a big thank you again to everybody at the New Brunswick Museum, all the people who help collect, obviously the New Brunswick Wildlife Trust Fund that gave us the funds to do most of the molecular work that you have seen here, all the people who send collections for the ongoing projects. And you can see here, I'm an ecologist in training. This is one of my cats, Martin, that is still very interested to try to figure out what are all these mushrooms that I bring home. And I will be more than happy now to go through the chat and start answering your questions. Let's see. I'm gonna try to share the screen so you can see. Where's the chat? The question as I'm trying to go through them. Can you recommend a good guide for macrofungi in New Brunswick, preferably with keys? Keys are going to be tricky because, it's very hard to build them based only on morphology, but you have several field guides that are for sale at the New Brunswick boutique, or you can find them in any bookstore. The mushrooms are of Northeastern North America by Tim Baroni, and then Mushrooms of the Northeast by Walt Sturgeon, and I'm forgetting the name of the other co-author, I'm sorry. And there are a couple of also field guides in French from our people in Quebec that are also applied to New Brunswick. So look for those. When you talk about mycorrhizal partners being nearby, what distance is nearby? We try to record the tree species the mushroom was directly collected under. And we make notes of anything that is nearby. For example, many of our collections will have notes collected directly under fir with birch nearby. Can I tell for sure if the mushroom is with the fir or with the birch? No, but that's the best we can do without getting into mycorrhizal studies. And do you have a New Brunswick focus key for these species? No, but hopefully I will build one soon. Once the new species of Hydnum is out, at least I will try to build a PDF of the species we have in New Brunswick. How common is hybridization in this macrofungi? Good question. I would skip over this question because it gives me a headache. I mentioned the study by Rachel Sweeney on chanterelles, and I highly recommend that if you're interested in hybridization, you check it out, because it really tells that some of these molecular lineages might still have contact with each other. There is a lot of, if you're interested in hybrids in macrofungi, look for the work of Dr. Karen Hughes of also the University of Tennessee. She has done amazing work trying to figure out the history and the possible hybrid history on macrofungi. I don't think it's extensively common, but there are definitely groups where hybridization happens. But yeah, and you can send me an email anytime, and I'm more than happy to give you more references for hybridization. Great talk, thank you. How can we collect in a way that you can use the specimen if we live far from St. John? There are some notes in the Chanterelles of New Brunswick project page and the Cortinarius of New Brunswick project page that gives you detailed instructions on how to collect for science. The main thing to collect, if you're gonna keep the specimen for science, is take good photos in the field, make a good note of where the species was growing, what type of forest or habitat, and then dry the specimen. Those are the three things that I would say they're basic, and that is already the specimen that is going to be useful for science. Do you have more than one species of Cortinarius semisanguineus, and what do you call it now? Yes, we do. I would say semisanguineus in the strict sense does occur in North America, but it's much, much rarer than other species. There is another big common species of semisanguineous looking that is either Cortinarius ominosus or Cortinarius tinctorum. In the catalog from Quebec, they use Cortinarius tinctorum. That might be the name in the end. Ominosus is actually older, so that might be the name in the end, but that is probably the most common one that we have here also. We also have Cortinarius cruentifilus, which it also looks like semisanguineous. I don't remember from memory if there are differences in terms of spore size or habitat that could allow you to tell them apart. DNA-wise, they're different for sure. So again, we do have semisanguineous in North America, it's just not common. The most common one is the clade that in the Cortinarius of Quebec, they call tinctorum, and we'll see if that is the name that sticks out in the end or not. Thank you, thank you, thank you. How habitat specific are these groups? Not so, some of them are. Quebecense, Atlanticum, you're only gonna find them growing in moss cover areas, bryophyte cover areas. Again, it doesn't have to be sphagnum. It's commonly sphagnum, but there are other, as long as the patch is, that to me indicates that they prefer really humid spots to fruit. And yeah, so they're not super picky in terms of both Chanterelles and Hydnums are mycorrhizal. Cantharellus and Alensis seems to prefer conifers like fir, spruce, but we also recorded it from birch. Hydnums in general, they love conifers, but they also establish mycorrhizal partnerships with hardwoods. So they're not super picky in terms of mycorrhizal partners. They might be pickier in terms of other abiotic or determinants of the habitat that for some of them haven't quite figured out yet. But yeah, I would say for Hydnums and Chanterelles, they're not super picky, at least in terms of mycorrhizal partners. Yeah, Damien, don't worry, it wasn't a difficult question. It's just hybridization is such a, in general in biology, and especially for people who do taxonomy, we don't want to think of, we taxonomists like to put things into neat boxes, and hybridization messes up those boxes really badly. So we don't like to think about that, but it does occur, and it's very interesting and it's fascinating, but it's not something I like to think about a lot. Thanks everybody for the, I don't think What's your favorite edible New Brunswick mushrooms? All hedgehogs. All hedgehogs are my favorite. They're all great. Hydnum atlanticum, by the way, tastes fine. It's edible. We tried it. All hedgehogs are probably my favorite. What led you to being so passionate about this specialized work? When you're a biologist, I did my undergrad in biology, and at some point, if you're going to go into research, you have to narrow down what you want to do. Biodiversity was always a target, and I had an excellent professor of mycology on my third year as an undergrad, and she later became my PhD advisor. So having good mentors makes a whole difference. And also fungi are the most fascinating organisms out there. Like animals and plants are boring. I'm sorry for everybody passionate about animals and plants. Fungi are still the best. Do you prefer mushroom observer or iNaturalist for posting mushroom finds? iNaturalist is more active. I still check mushroom observer, and I think they have done a great job. I like iNaturalist better. Everybody's on iNaturalist, really. The mycological community has kind of migrated a lot towards iNaturalist versus Mushroom Observer. I still use Mushroom Observer. I still try to go there, suggest ideas anywhere I can. And yeah, I think that's it for questions. I see Mary now. I'm going to stop sharing my screen. So.
Okay, well, you have to excuse me, the snowplow's going by, so I hope that's isn't too much in the background, but thank you so much, Alfredo.
Yeah.
That was a really great presentation, and you know, you put up all these scary looking clades and you make it seem so accessible to us, so thank you for doing that. And also, it's great that you really imparted the importance of the collaboration between scientists and amateur collectors, and also the importance of the sequencing to really know what species you have out there. And it's something that a lot of us we just want to look at them from the outside and figure out what they are. But yeah, you've made it really obvious that the sequencing is an important part. So congratulations on all the discoveries that you've made so far. And it sounds like you still have a lot to find and a lot to name. Thank you so much, Alfredo, for that.
Yeah, thank you for having me. And thanks everybody for the great questions.
Yeah, a great audience today. So I will just mention that our next guest speaker will be Saturday, March the 18th, and that will be Adam Cheeseman discussing nature and bee projects in the St. John area. And that will be on Zoom. And don't forget to check the St. John Naturalist website and Nature St. John Facebook page for upcoming events so that you can register. So thank you, Alfredo. Thank you, everyone, and have a wonderful weekend. Bye.
Thank you. Bye.